So I hope we have to start, and the topic for our today's class is inflammation. It is really a great topic, and it is really important to understand the mechanism of development of inflammation, because a lot of inflammatory disease we will discuss a bit later, and all of them are really important to understand the mechanism of the general process like inflammation. And inflammation, one of the biggest part of pathology, and today we will discuss a lot of different types of it, and all of them has some special manifestations, and all of them has special causes for development. So let's please start. First of all, we should start from the definition of this pathological process. So inflammation, it is a complex, local, vascular mesenchymal reaction of an organism aimed at the destruction of causative agent and tissue structure recovery. So here we may note that the inflammation localized in the vessels and in stroma of organs, and it is the result of injury, and the most important idea and most important meanings of inflammation is destruction of these pathological agent, and in the end of inflammation we should see structure recovery of these tissues. There are a lot of different etiological factors of inflammation. They could be divided in several groups. For example, it may be biological factors, chemical factors, and physical factors. Also, we may see a lot of other different types of factors or inflammatory factors. It also could be endothelial factors, for example, like autoimmune reactions, and a lot of other factors. But the most important you may see on this slide. In biological factors, we may see viruses, and you know that a lot of different diseases could be associated with viruses. One of the most important in nowadays is coronavirus infection, and where typical changes which happens in the organism is associated with inflammation, and we will discuss a bit later special type of these infections. Bacterial infections are also may be associated with inflammatory reaction of the organism, and we will see special manifestation of inflammation here. Fungus also, animals, parasites also, but they will have some different type of inflammatory reaction. So circulating in blood antibodies and activated immune complexes or autoimmune factors of alteration are really popular in population, and we may see a lot of autoimmune reactions in nowadays. Also, we should mark some chemical factors like acids, alkalis, minerals, and organic substances, and also sometimes we may see endogenous toxins like bile agents, like nitrogen metabolism products, like carbohydrate oxide, and all other chemical factors which could be contained in our organism. And also we may have deal with physical factors like traumas, like radiations, like electric energy, and all other, like it may be like hot and cold, and a lot of other physical factors. I should tell you one more time that on this slide I named only the most important and most common factors, but not all of them. There are a lot of different groups of these alterative factors which may lead to development of inflammatory reaction. All inflammatory reactions has same clinical things, clinical features, which may be associated with reddening or rubor, edema or tumor, temperature rise or color, and it may be associated with pain or function derangement or functional lesion. And on this slide we may see like a schematically manifestation of these factors in one image. So these 5 factors or these 5 manifestation of inflammation will be manifested in any case of inflammation. They are typical for all types of inflammation. Today we will discuss a lot of different variants of inflammatory
Excuse me,
reaction.
sir.
Yes.
Sir, I cannot see your PPT.
So you didn't see my presentation, yeah?
Yes, sir.
Okay. And what can you tell me about it right now?
Yes, sir, now we can see.
Uh-huh. Thank you for your notes. Uh, so on this image you may see scheme of clinical things of inflammation. Please, just for understanding and for your mention, for your memory, it looks like this. There are 3 stages of inflammatory reaction, and they are alteration, exudation, and proliferation. These 3 stages are present in all different types of inflammation, but in different kinds of it, some stages prevalence under all others. And uh, the first stage of alteration, and the first stage we have already discussed on our previous classes when we discussed dystrophies, when we discussed necrosis, when we discussed some blood circulation disorders. All of these problems manifestate itself like alteration. So here we may see damage of our tissues, and the damage of our tissues will lead to uh, activation of inflammatory process in our bodies. Parenchyma and stroma degeneration up to necrosis. Release of bioactive substances are important manifestation of this stage, and the most important meaning of this stage is release of bioactive substances. So in the end of alterative stage we may see uh, accumulation of a lot of bioactive substances in the area of damage, and these bioactive substances will lead to activation and progressive of inflammatory reaction, and all of these substances will have influence to outcome of inflammatory reaction. And in the first stage of inflammation we may see a prognosis of inflammatory reaction. And it is important to note that any inflammatory reactions begins with alteration stage, and inflammation triggering mechanism is release of bioactive substances. So I should tell it one more time that release of bioactive substances is the most important idea or the most important meaning of alteration stage. The sources of bioactive substances may be different, and I hope you have already discussed it in the pathological physiology department and in your classes about pathological physiology uh, classes. But I should revise you some places where these substances could be accumulated. And first of all, it may be plasma bioactive substances, for example, like chirocrene system, complement system, or blood coagulation system. And also, they may be contained in the area of inflammation or cellular factors cellular inflammatory factors or local inflammatory factors, which could be contained in the labrocytes, thrombocytes, basophils, macrophages, lymphocytes, fibroblasts, neutrophils, and all other cells which could be involved in the inflammatory process. And we should pay more attention for these factors because they have a great influence to development of inflammatory reaction. So in cellular factors, we may note histamine and serotonin, bradykinin, blood coagulation system again, and here we should pay more attention for factors which are associated with thrombocytes, for example, fibrin, neopeptides, complement systems for C5a and C3a, one of the most important factors of inflammation, and proteases of neutrophils, which have great influence to the suppurative inflammation, and neutrophils could be one of the most important protectors in case of bacterial infections, for example. And in the result of action of all bioactive substances which accumulates in the first stage, we may see the development of the second stage of inflammation, the stage of exudation. It is a complicated process of effusion formation, and there are two the most important parts of this effusion or exudate. It may be liquid part which contain water and proteins, and also it contain a lot of cells and all other particles of our blood. In the stage of exudation, we should note two phases of it. The first phase is microcirculatory changes which associated with stasis, which associated with blockage of the bloodstream in the periphery of alterative area, and also we will see cellular reaction. And cellular reaction most of all associated with migration of leukocytes, migration of fibroblasts, migration of macrophages, and all other cells of the blood. So we will discuss it a bit later and more closely. So microcirculatory changes associated with short-term vasoconstriction in the periphery of alterative area. So when we will see damage, our organism tries to make a border of this damage, and that's why in the periphery of this pathological area we may see vasoconstriction or spasm of small arteries. After that, we will see vasodilatation and stasis in the middle of this alterative area. So we will see blockage of vessels in the periphery and vasodilatation and stasis in the middle of this center. It is necessary for hyperemia here, and in our previous class we have already talked about inflammatory hyperemia, and inflammatory hyperemia happens in this moment. Increase of vascular wall permeability cause of like a result of vasodilatation and stasis, and here we will see increasing of fluid transition from microcirculatory system into the tissues. So here we will see accumulating of a lot of fluids in the intracellular space, and this accumulation of fluids is necessary for normal activities of cells, of inflammatory cells, and they are really important for initiation of inflammation at all. Several reactions associated with marginal or age standing of leukocytes at the first stage. After that, we may see active migration of inflammatory cell from the blood into lesion area in the area of damage from the blood vessel, and here we may see transport of this inflammatory cell from blood vessel into the damaged tissues. After that, we will see phagocytosis, and phagocytosis is a process of feeding or elimination of alterative agency, and it is really important to understand that it is really necessary to have a lot of fluids, to have a lot of bioactive substances in this area for normal activities in this stage. And in the end of this process, we will see formation of exudate. Exudate, it is accumulation of fluids in the tissues or in our organism, and we could see accumulation of fluids in our organism in case of two different processes, in case of transudate and exudation, So, and now we should discuss the difference between transudate and exudate formation. The transudate is characterized by non-inflammatory reaction. For example, it may be associated with heart insufficiency, with ischemical heart diseases, or something about it. Exudate, it is inflammatory liquid. It is typical only for inflammation. And on this slide you may see the difference between transudate and exudate. The difference between them associated with some different components of these liquids, different causes of their formation, and different mechanism of its formation. So you may see it on the slide, and I will now read this slide in the lecture, so you may see, and please remember these factors. And after the stopping of second stage, we will see formation of the third stage of proliferation. And the main idea of proliferation stage is recovery of damaged tissues. And the most important cell of this reaction is fibroblasts. And the most important idea of this stage is recovery of damaged tissues, and most of all it will be associated with synthesis of connective tissue and replacement of damaged tissue to connective one. So on the proliferation stage we may see increase of cell quantity in the inflammatory zone of the local histogenous and hematogenous origin. And it is really important to remember some histogenous and hematogenous cells. In histogenous cells, we should pay attention for epithelial cells and fibroblasts. In hematogenous cells, we may see a lot of different kind of cells. It may be monocytic layer. For example, it may be monocytes, macrophages, epithelioid cells, and Giant cells. It may be lymphocytes, it may be B lymphocytes, labrocytes, and plasma cells. It could be neutrophils and T lymphocytes. And so please remember these cells like an example of hematogenous and histogenous cells. Regulation of inflammation based on three most important factors: on hormone regulation, in neural regulation, and immune regulation. The hormone regulation associated with pro-inflammatory hormones like mineralocorticoids, somatotropin, hypophyseal tyrotropin, aldosterone, and some other hormones, and actions of anti-inflammatory hormones like glucocorticoids and adrenocorticotropic hormone. In the balance of actions of these two groups of hormones, we will see activation or stopping of inflammatory process. And also it is really important to know the group of hormone if we try to treat the patient by these hormones, because sometimes we will use some hormones for activation or for decreasing of the inflammatory reactions. In neural factors, we may see also pro-inflammatory factors and anti-inflammatory factors. Pro-inflammatory factors like cholinergic substances will stimulate the output of inflammatory mediators, and anti-inflammatory factors, like an example, we will see adrenergic substances depress the activity of mediators like anti-inflammatory hormones. So the most important idea of this classification is theoretical, probably theoretical methods of classification of some factors which could have influence to the inflammatory reaction. And one of the most important regulation factor are immune factors. And the immunity state has a great influence to intensivity of inflammatory reactions. Sometimes we will see hyperergic or hypoergic reactions in different kinds of immune system status. Most of all, probably you know about some patient with HIV infection, and we may see that it is not typical to have a great inflammatory reaction for them. Sometimes we will see a great immune activities in patient, and it may lead to formation of some special types of inflammatory reaction, for example, to formation of granulomas, and we will discuss it a bit later. So the main idea, please remember the three most important factors which may have influence to the inflammatory reaction, and just understand when in each of the group we may see factors which may increase the inflammatory reaction, and also we can see some factors which will lead to depress of this inflammatory reaction here. So it was all about the theoretical part of general inflammation, and now we should discuss the special type of inflammation. So according to course, inflammation could be classified to acute, subacute, and chronic. Acute inflammation would take something about some hours, subacute something about 3 months, chronic more than 3 months. Inflammation forms according to morphology, and one of the most important type of inflammation is classification according to morphology. We have only two types of inflammation, exudative and proliferative one. The main idea is the prevalence of the inflammatory stage. Exudative inflammation. In case of exudative inflammation, we may see that exudation stage prevalence on other alteration and proliferation with the reaction of microcirculatory bed in the exudate formation. So I should pay your attention that all other stages like alteration and proliferation are present in case of exudative inflammation, but the most important influence on the morphology has exudative form of it, or exudative stage of it. And composition of exudate you may see on this slide. So exudate contain fluid, exudate contain proteins, it contains uniform elements of the blood, cell of local tissues, decay products, and sometimes it could be microbes or foreign bodies or viruses or something from the alternative agency. The exudative inflammation could be divided into different groups, and the most important of them are independent and dependent types. So the difference between them: the independent types could be a single type of inflammation. So we could see only serous or only fibrinous or only suppurative type of inflammation. But the depends type of inflammation must be associated with independent types. So sometimes we will see Catarrhal inflammation in case of Serous Catarrhal 1, or Serous Hemorrhagic, or Fibrinous Putrid inflammation. And also we should remember about mixed type. Mixed type of inflammation, it is a situation when we may see a combination of two of independent type. So it could be serous fibrinous inflammation or serous suppurative or fibrinous suppurative type of inflammation. And sometimes we may see accumulation of two of independent and one of dependent. And also, it is really important to pay attention for Catarrhal type of inflammation, because it is one special type of inflammation which could be localized only on mucous membranes. And in any case, when we see inflammatory reaction on the mucous membrane, it should begins from the Catarrhal inflammation. So Catarrhal inflammation will be the first stage of damaging of mucous membrane. So that's why it could be except from the dependent types and put to some other types. So in the classification methods of exudative inflammation, sometimes we put Catarrhal inflammation in the third group, in the group of Catarrhal inflammation, because it is a special type of inflammation which associated with localization of a process. All other types are associated with containing of different products in exudate. So, and now we should discuss the special anatomical manifestation of these types of exudate. So, serous inflammation could be localized on mucous membranes and serous membranes, lungs, meninx, vasculosa, and skin. And the composition of this exudate is fluid, plus plasma proteins and single cells, and it one of the most favorable type of exudative inflammation, because here we will see full recovery of damaging tissues with exudate resorption. The color of exudate could be different. It could be semi-transparent or transparent, absolutely transparent, but sometimes it may have some whitish color or yellowish color. And here we will see the morphological manifestation of a process. Histologically, we will see some empty spaces between cells, and which are associated with accumulation of a lot of fluids here. And morphological, macroscopically, we will see something about like bubbles, or if it's localized on the skin. If it localized on the mucous membranes, we will see just a lot of fluids on the surface. And if we see the serous inflammation in the lungs, we will see a lot of fluids in the lungs with some gray color, for example. The fibrinous inflammation. The fibrinous inflammation it is localized on the mucous and serous membranes, and sometimes we may see it in the lungs and some other internal organs. And it has two forms. It has crupose and diphtheritic form. And the most important to understand the difference between crupose and diphtheritic type of inflammation. The crupose inflammation associated with formation of like hairs on the surface of the organs. It is associated with superficial necrosis of underlying tissues, and it is associated with formation of exudate with gray or white color, and it is localized only on the single layer prismatic epithelium. The diphtheritic type of inflammation most of all localized in the areas with multiply epithelium, and it characterized by the deep area of necrosis and with huge accumulation and huge association between the exudate and underlying tissues. The different outcomes of the fibrinous inflammation are possible. It could be associated with exudate resorption, mostly in case of Kupol's inflammation. In case of Kupol's and diphtheritic, in both types of fibrinous inflammation, we may see organization of exudate. We could see commissures formation, like on this slide we may see hairy heart, and hairy heart is associated with development of commissures between the heart and pericardium, and on the surface of the heart we may see a lot of short white fibrils, which looks like hair, and that's why the heart has name high heart. Epithelialization of the damaged area and formation of ulcers under the inflammatory fluid. Suppurative inflammation. Suppurative inflammation, it is a great form of exudative inflammation, and it is really aggressive form of it, because here we will see purulent bodies, and purulent bodies, it is a basis of this exudate, and the morphological substrate of it is dead neutrophilic leukocytes. And you know that these cells accumulate a lot of aggressive fluids in them. So there are a lot of different enzymes inside of them. And that's why the sap or exudate, purulent exudate, contain a lot of enzymes which could lead to destroying of surrounding tissues. And it may make a lot of troubles to organism and to the patient. The localization of suppurative inflammation is really different. It may localize in the all tissues and all different localization. And morphologically, you should know 4 forms of it. They are abscess, phlegmon, MPM, and apostema abscesses. It is really too important, the difference between them. So on this part of the slide we may see abscess. So abscess, it is a border or suppurative inflammation with borders. The sap is accumulates in special cavity which created in result of inflammatory process, and sometimes we may see it in different cases. For example, here it may be a result of accumulates of foreign bodies in the skin. It may be a result of some inflammatory process in the for example, abdominal cavities and formation of under diaphragmal abscesses, and something about it. They may localized in any places, but the main idea of abscess is formation of a border, and this border will protect organism against the enzymes of sap, and the borders will protect our organism against the sap. The second form is phlegmon. Phlegmon is suppurative inflammation without borders. Here we may see spreading of inflammatory reaction between soft tissues and in huge area. For example, here we may see phlegmon of the neck, and the typical reason of it is a caries of teeth, and it may lead to inflammatory process, suppurative inflammatory process of the neck, and may localize in the mediastinum, for example, and may lead to problems with the heart, with problems with the thorax, and could be spread on all areas under the neck. The third form is empyema. Empyema, it is accumulation of a sap in normal cavities of the body. For example, here we may see empyema of pleura, so it is accumulation of sap in pleural cavity, and this process may localize not only in the pleura but in all organs of our organism with cavities. For example, it could be all bowders. For example, it may be gallbladder or urinary bladder. It could be gastrointestinal tract. It could be uterus. For example, it may be, for example, empyema of the pericardium and all other cavities of our body. And apostema abscesses. Apostema abscesses, it is a form of suppurative inflammation which morphological closely to abscess, but it has some special characteristics. So in case of abscess, we may see only single formation, and most of all it has a huge size. In case of apostema abscesses, we will see a lot of small pointed disseminated abscesses in organism. It is typical for generalized infections. For example, it's typical for sepsis and the formation of generalization of suppurative inflammation. So on this slide we may see apostema of the kidney, and we will see these yellow dots in the kidney, and all of these dots has size something about 1 or 3 millimeters in diameter, and all of them contain a lot of sap. So I hope you understood the difference between different forms of suppurative inflammation. Hemorrhagic inflammation. The basis of exudate is erythrocytes, and on the surface of injured tissues we will see red masses, the tissues becomes red and contain a lot of erythrocytes in case of histological examination. This type of inflammation is interesting because in case of infectional agency we may see it in some dangerous infections like plague, anthrax, influenza, coronavirus, and Ebola virus. So we will see this difficult type of inflammation. What is the common characteristics of all of these infections? All of them will lead to dramatically increase of vascular permeability. Some of them will lead to damage of vessels, microcirculatory vessels, and like a result of that, we will see a lot of erythrocytes in the exudate. And that's why they are really dangerous for people. Putrid inflammation develops by the joining in result of a joining putretic microorganism or Clostridium. It could have different localization, but the most important characteristics of it is deep wounds or some areas of our organism without output of the oxygen, because for development of Clostridium and for their life cycle is really important to some conditions without oxygen. That's why this type of inflammation could be localized only in these places. One of the typical characteristics of this exudate, the exudate fluid will looks like Serous or Fibrinous type of inflammation, but the special feature will be associated with disgusting smell, and smell will looks like will have the smell will be associated with formation of hydra hydrosulfatis, and the smell will have really disgust. For example, like old eggs in refrigerator. For example, if you forget the eggs in the refrigerator more than 1 month and try to open it and try to make an breakfast from this egg, you will smell this disgusting smell. And catarrhal inflammation. Catarrhal inflammation localized only on the mucous membranes, and exudate contains a lot of carboproteins which have name mucus. And the different subtypes of catarrhal inflammation could be serous catarrh, mucous catarrh, and purulent catarrh. It is the development and the changes of these types of catarrhal inflammatory exudate you may see in the example of runny nose. In first 2 days of runny nose, we may see serous catarrh. It will be fluid without smell, and it will be fluid, semi-transparent or glass-like fluids, and in few days it becomes white and sometimes or milky color, and it will be mucous catarrh. This fluid will have some sweet smell, and it will be associated with accumulation of carboproteins in it. And after 1 week, we may see exudate with green color. It will be purulent catarrh. So it will be associated with addition of some biological agents like microbes, and in result of it we will see purulent inflammation here. So it was all about exudative forms of inflammation, and now we have to discuss the productive inflammation. The productive inflammation, it is a special form of inflammation where the proliferative stage is prevails upon alteration and exudate. And again, I should pay your attention to fact that alteration and exudation stage are also present, but the proliferation stage is prevalent on others. Product inflammation mostly has chronic course, but sometimes it may be acute in case of some viruses and other diseases. We will discuss it a bit later. And acute course may be observed in case of several infections like typhoid fever, spotted fever, rabbit fever, brucellosis, and infectious agency diseases like acute rheumatism, acute glomerulonephritis, and something about it. In case of productive inflammation, the stage of proliferation is the main stage of inflammation, and in this stage I have already told you in the beginning of this lecture that we may see a lot of cells in the inflammatory area, And here we will see a lot of monocytes which could be transformed to macrophages. We will see lymphocytes like T and B lymphocytes, and in result of their development we will see plasma cells. We will see eosinophils, we will see labrocytes, we will see leukocytes, fibroblasts, and epithelioid cells like a product of macrophages. Types of proliferative inflammation. There are three most important types of it. They are interstitial, inflammation with formation of polyps and pointed condylomas, and granulomatous type of inflammation. And we will discuss them a bit closer. The interstitial inflammation could be localized in the parenchymatous organs like liver, kidney, myocardium. It may localize in the lungs, brain, and all other parenchymatous organs. Organs appearance practically without changes, so macroscopically it is really difficult to see the changes, the morphological manifestation of this inflammatory reaction. Histologically, we will see some inflammatory cells between parenchymatous cell of the body, and we will see histiocytes, lymphocytes, macrophages, eosinophils, labrocytes, and single leukocytes between normal cells of the organ. And like action of these cells and like a result of inflammatory reaction here, we will see dystrophic and necrodybiotic processes in surrounding parenchymatous cells. And most of all, this process will lead to overgrowing of connective tissue, and we have a special word for overgrowing of connective tissue in case of inflammatory reaction. It will be sclerosis. So on this slide we may see histological picture of interstitial inflammation. So here we will see cardiomyocytes here and here, and between them we will see a lot of inflammatory cells. So I hope you can see it. The small blue dots between pink masses. These pink masses are cardiomyocytes, and the cells between them we are lymphocytes, labrocytes, some macrophages maybe we may see in this area. And same picture but closer. Probably it is this area should be, and we will see that in muscles tissue we will see some manifestations of necrosis, like here we can see cells without nucleus. We can see some cells in degeneration process and something about it. In the inflammation with formation of polyps and pointed condylomas, so in case of this inflammation, we will see the overgrowing of stroma, and which could be covered by two different types of epithelium. It could be prismatic epithelium, and it could be multiplier flat epithelium. In case of prismatic epithelium, the overgrowing of stroma will have name polyp. In case of flat epithelium, it will be condyloma. The typical causes of this inflammation will be associated with some viruses or chronic inflammatory reaction in places of changing of one type epithelium to another one. It will be associated with inflammatory process in some areas like lips, like I don't know, mucous membranes, for example. And it is really we should pay attention for this formation, because sometimes the polyps and condyloma may lead to formation of malignant tumors. That's why we should pay attention for these formations in the organism. And one of the most interesting type of proliferative inflammation It's granulomatous 1. The granulomatous inflammation, it is a special type of proliferation inflammation which is associated with formation of special nodes, granulomas. And granuloma or nodule, it is a focal cluster of a cell which have ability to phagocytosis. So we could see a lot of different diseases with formation of granulomas. And granuloma could be classified to special and non-special one. We will discuss it a bit later. First of all, we should discuss the morphogenesis of it. The morphogenesis or stages of granulomas formation you may see on this slide. I try to explain it more easier that this process is associated with development of monocytes, and you should remember the ways of transforming of monocyte. The first of all, monocyte could be transformed to macrophages. Macrophages could be transformed to epithelioid cells, and accumulation of epithelioid cells will lead to formation of giant cells. I hope it is easier to remember the ways of transformation of one cell, monocyte. Monocyte, macrophages, epithelioid cell, giant cell. In the granuloma we will have same stages. So it will be formation of monocyte granuloma, formation of macrophageal granuloma, formation of epithelioid cell granuloma, and in the end we will see formation of the giant cell granuloma. Classification of granulomatous disease you may see on this slide. So we will see granulomatous disease with infectional etiology. We will see granulomatous disease with non-infectional etiology. We will see drug-induced granuloma and granulomatous disease with unknown etiology. And it is really interesting for us that granulomas with infectional etiology could be divided to specific and non-specific. The specific infectional granuloma, it is granuloma which has special morphological picture, and in examination of this morphological picture we could make a diagnosis. So limited productive inflammation with formation of granuloma with structures and composital are typical for certain form of the causative agent. For example, we will see some anatomical formation, and only in this picture we may associate it with special disease. I will give you examples a bit later. They have some special characteristics, like each of them has a strictly defined pathogen, chronic adulting course, changing of tissues reaction during the inflammation, development of a granuloma with definite structure, and characteristic is the development of a caseous coagulative necrosis. So, special infectional granuloma could be associated with TB infection. And in TB infection, we will see tuberculum node. In the center of this granuloma, we will see area of caseous necrosis. On the necrosis zone periphery, we will see epithelioid cell, lymphocytes, and gigant cells, which we may see here. Gigant cell of Pyrgo Langhans. Here it is a special cell with nuclear accumulation in form of the arc on the periphery of a cell. The second special granuloma, it is a granuloma in syphilis. The name of it is guma. In the center we may see colliquative type of necrosis, and we will see a lot of plasmacytes, lymphocytes, epithelioid cells, and sometimes we will see here Pirogov-Langhans cell. Macroscopically, the guma has size something about 2.5 centimeter, 25 millimeters, and up to 5 centimeters. And in the outcome, we will see scar in this formation. It is interesting to note that the guma could be formed only in the tertiary period of the syphilis, and it develops after the 10 or more years after the infected of the patient by the syphilis. In case of TB infection, macroscopically, the size of the tuberculum node is something about 1 or 2 millimeters, and it could be localized in any organs. It could be localized in the bones, it could be localized in the kidney, in the lungs, and all other in all other localizations. In case of leprosy, the name of granuloma is lepron, is replaced mainly by macrophages as well as plasmacytes and lymphocytes, and specific cells are Virchow cells. We will see it here. It is a cells with special pink masses inside of them. And non-specific granulomatous inflammation, it is granuloma which are typical for a lot of different types of diseases. For example, they could be found in case of rheumatism, in typhoid fever, in typhus, in actinomycosis, aspergillosis, inflammation around parasites and foreign bodies. For example, granuloma in case of rheumatism, it is a Ashoftal lymph node, and we may see it on this slide. In the center, we will see area of fibrinoid necrosis with unstructured masses here, and around it we will see cluster of macrophages with high cells. So sometimes we may see Pirogov-like cells, like here, and all other giant cells. So, outcome of granulomatous inflammation, most of all, is formation of scar, formation of connective tissue in place of the pathological process, and the size of scarring will be associated with the size of granulome in different diseases. Importance of the granulomas could be associated with and mostly typical for specific type of granulomas. Sometimes it may make a diagnosis only by morphology. We may identify the cause of this disease. Sometimes granuloma is a reflection of immune state of an organism, and granulomatous inflammation is typical only for strong immune system, because it is a result of huge activity of immune system here. And it's all. Probably it's all theoretical part for today. Thank you for your attention. I was waiting for your question. Goodbye.